I would like to welcome next presenter, Dr. Archana Manerikar. Dr. Manerikar serves as a bioequivalence assessor in the Office of Generic Drugs. Dr. Manerikar has experience in assessing topical drug product applications. Her presentation title is Considerations for IVPT Data and Information Submitted in ANDAs.
Thank you, Dr. Katraga. Is everyone able to look at my slides?
They look great, yes.
Okay, thank you. Thank you, Dr. Katraga, for the kind introduction. I'd like to begin by thanking the workshop organizers for this opportunity to present to you today. Now that we've looked at the submission considerations for IVPT studies as shared by Dr. Ma, let us discuss some important considerations that are relevant to IVPT studies submitted in ANDAs. Kindly note that this presentation reflects my own views and should not be construed to represent FDA's views or policies. In the next 27 minutes or so, I will attempt to cover goals of today's presentation, considerations for how to organize information and data in the IVPT submission, submission considerations that are relevant to all IVPT studies, submission considerations that are specific to various IVPT studies, and then conclude with a brief summary. In this presentation, the specific submission considerations have been classified into two categories. Those relevant to the IVPT method, that is, considerations pertaining to the method development, method validation, including pilot and pivotal studies, and those relevant to the analytical method, that is, considerations pertaining to the analytical method validation studies, as well as to sample analyses for IVPT studies. So in discussing important considerations of information and data to be submitted along with their organization within the IVPT submission, the goal is to help generic applicants enhance the quality of their IVPT submissions and to facilitate a reduction in the number of deficiency comments as well as the number of ANDA assessment cycles. Now in the next slide, we'll briefly touch upon how to organize information in an IVPT submission. Now, in general, summary information pertaining to IVPT studies may be submitted in Module 2.7. We recognize that it may be helpful to applicants if FDA were to provide details of specific IVRT and IVPT study design and data elements that facilitate assessment of such studies submitted in ANDAs. Therefore, we look forward to receiving your feedback regarding the potential utility of, as well as suggestions for, such summary tables that FDA could prepare. Now, moving to Module 5, detailed information and data relevant to the IVPT method, that is, details of the method development, method validation, including pilot and pivotal studies, may be submitted in Module 5312. Examples of such information and data may include study reports, data sets, data definition files, standard documents such as protocols, standard operating procedures or SOPs, standard testing procedures or STPs, along with any additional supporting information. Similarly, data and information relevant to the analytical method, that is, details of the analytical method validation studies, as well as details of sample analyses for IVPT studies, may be submitted in Module 5314. Examples of such information and data include study reports, numerical raw data, representative chromatograms, standard documents, such as, for example, protocols, SOPs, and STPs, and any additional supporting information. Over the next 6 slides, we will look at some general submission considerations that are relevant to all IVPT studies. Across the IVPT submission, please consider submitting detailed, well-organized, and separate study reports for IVPT studies, such as method development, validation, pilot, and pivotal, for analytical studies, as well as for sample analyses relevant to the method validation, pilot, and pivotal studies. In the IVPT submission, it is important that experiments and results that were part of the method development are clearly segregated and identified as being distinct from those that were part of the method validation. Consider providing all standard documents as relevant to the IVPT method and study conduct, as well as to the analytical method and IVPT sample analysis. For each standard document, consider providing detailed summary information such as document number, title, brief description, effective date, validity period as applicable. Also, consider ensuring that the submission includes adequate demonstration of consistency and control for all relevant study procedures and method parameters over the IVPT study duration, such as those pertaining to skin, for example, preparation and mounting of the sections, conditions of skin storage, measurements of skin thickness and integrity, pertaining to receptor solution sampling, such as, for example, sampling time precision, sampling technique, duration of sampling and replacement of receptor solution, sample volume or flow rate, and sample handling and storage. Also, those pertaining to dosing, such as, for example, area of dose application, actual dose amount applied, dosing technique, as well as dose duration. Those pertaining to measurement of pre-dose concentrations, which, as we have heard over the discussions over the last 2 days, can help identify potential contamination associated with each skin section and or each cell. And finally, pertaining to the use of control cells with non-dose skin sections, which can not only assist with the periodic monitoring of temperature at the skin surface and at the receptor solution over the study duration, but can also help ensure that the drug concentrations observed in the receptor solution are in fact associated with the applied dose and not with any drug contamination in the skin. Now, for product lots used in the IVPT studies, consider providing details such as lot number, manufacturing date, batch formula and size, product strength, potency and content uniformity if available. If altered formulations are used, explanation of the specific alteration that was made along with its rationale, as well as description of any altered manufacturing processes. And please consider providing expiration dates for lots of commercially available products that were used. Pertaining to each skin section used in the IVPT studies, consider providing details such as donor identification, supplier information, anatomical site of skin collection, donor demographics, along with individual values, mean, and precision for both skin thickness measurements as well as barrier integrity test results. Pertaining to the handling of each skin section as used in the studies, consider providing details such as, for example, conditions of sample receipt from the vendor, conditions of storage, time and date of each instance of retrieval from and restorage back in the deep freezer, along with the number of freeze-thaw cycles that are relevant to each skin section. From an assessment perspective, it is extremely helpful when such product lot and skin section details are provided separately for each IVPT study within your submission. Across all IVPT studies, consider providing a detailed discussion of experimental observations that may potentially influence the interpretation of the study results, as well as of any protocol or SOP deviations. It is very important to note that the submission of chronological, stepwise details of each IVPT procedure along with relevant data and calculations, represents a critical piece of information that helps assessors reconstruct how each IVPT study was conducted and verify each reported result. With that, let's proceed to some specific considerations for what to submit for each of the different IVPT studies. So over the next 3 slides, we will look at some specific submission considerations that are relevant to the IVPT method development studies. Alongside detailed, well-organized information that is submitted in the method development report, consider providing summary details relevant to each method development study, which pertains to data elements such as study information, such as study number, title, study and analytical dates, the report number and location, molarity pH of the receptor solution, information about sampling time points, sampling volume, study duration, skin source, anatomical site of the skin collection, skin thickness, barrier integrity test type, barrier integrity test instrument as used, as well as temperature maintained at the skin surface, as well as the number of donors and replicates used, the diffusion apparatus that was evaluated, along with dose amount, dose application method, and the dose duration as relevant to each method development experiment or study. For each method development study, also consider providing the range, mean values, and precision for Jmax, as well as for the total cumulative amount of drug permeated over the study duration. For each method development study, also consider providing mean profiles and tables containing mean values, standard deviation, and precision for flux and the cumulative amount of drug permeated at each sampling time point. For the final IVPT method, consider providing details of the drug solubility and the barrier integrity test acceptance criteria. In general, a method development report should provide detailed and well-organized documentation of all systematic efforts that were made to optimize the IVPT method. This may be accomplished, for example, by including detailed information for each method development study, by clearly indicating the specific method parameters, experimental conditions, and controls that are relevant to each set of data, and by clearly identifying, for example, based on unique cell identification, the specific diffusion cells that were run in parallel in each IVPT run involved in these studies. Overall, information and data submitted in the method development submission should assist in the understanding of why specific IVPT method parameters and procedures were utilized, as well as whether the resulting IVPT method is suitably sensitive and reproducible. Now, over the next 6 slides, we will look at some specific submission considerations as they pertain to the IVPT method validation, including pilot and pivotal studies. For the method validation, pilot, and pivotal studies, in addition to detailed, well-organized reports, consider providing summary details pertaining to data elements such as study information like study number, title, objective, study and analytical dates, report number, location, as well as testing and analytical site information, receptor solution, molarity and pH, antimicrobial agent and its concentration in the receptor solution, drug solubility and stability in the receptor solution, study duration, sampling time points, sampling volume, and sampling procedure, along with data elements such as skin source, anatomical site, skin thickness, barrier integrity test type, integrity test instrument, integrity test acceptance criteria, as well as temperature, which was maintained at the skin surface for the duration of the study, dose amount, dose application method, dose duration, diffusion cell information such as type of diffusion cell, volume, orifice area involved, as well as the number of diffusion cells that were used, number of donors and replicates that were involved, stirring or flow rate, environmental control and sample storage conditions as applicable, as well as randomization details as applicable to the pilot and pivotal studies. As a reminder, drug stability is demonstrated at the highest relevant temperature in the receptor solution that is exposed to the underside of skin for the longest relevant duration. Sample storage duration, which is basically the duration from the first date of sample collection to the last day of sample analysis, should be covered by the validated storage stability. For each treatment or formulation as evaluated in the pilot study, consider providing intra-donor and inter-donor values of mean and precision for Jmax, as well as the total cumulative amount of drug permeated over the study duration. For IVPT sensitivity and selectivity studies, please consider providing flux profiles in tabular as well as figure formats. Please also consider ensuring that all submitted standard documents, for example, protocols, SOPs, and STPs, contain detailed, coherent, stepwise, and chronological description of each IVPT procedure, contains detail of how each IVPT procedure and each parameter will be consistently controlled, as well as details of how evidence of such control across the procedures and parameters would be monitored, recorded, and reported. In the IVPT method validation, including pilot and pivotal study submissions, consider ensuring that relevant method parameters, including skin source, anatomical site, and preparation, along with study conduct and control procedures, as well as relevant acceptance criteria, are consistent across the method validation, including pilot and pivotal studies. It is also important that all relevant method parameters and experimental conditions and controls that correspond with each set of IVPT results are clearly and consistently identified in the submission. As well as, it is important that adequate evidence demonstrating consistency of the procedures and control of the parameters is submitted. In the relevant documents, consider clearly indicating whether the method validation, including pilot and pivotal study results, were obtained using a validated analytical method. As part of apparatus qualification, consider identifying each diffusion cell as used in the method validation, pilot, and pivotal studies with a unique cell identifier, as well as consider providing, in a tabular format, empirical measurements of orifice areas and receptor solution compartment volume, along with any manufacturer-provided data, if available. Across the IVPT method validation, including pilot and pivotal studies, for each IVPT run, consider providing summary details, which include date of the IVPT run, diffusion cells identified by unique identifiers that were run in parallel in each IVPT run, identification of the dosed and the control cells in each run, treatment or formulation that was administered to each dosed cell in the run, as well as the number of the corresponding analytical run in which the samples were analyzed. Also, consider providing spreadsheets containing complete concentration data and relevant calculations for these studies. In these spreadsheets, clearly indicate the experiment to which each set of data belongs, as well as identify each diffusion cell with its unique cell identifier. Now, over the next 2 slides, we will take a look at the specific submission considerations that only pertain to IVPT pilot and pivotal studies. For IVPT pilot and pivotal studies, consider providing SAS transport datasets for concentration as well as PK data, along with respective data definition files that indicate relevant units. Examples of data elements that may be included in such SAS concentration datasets are provided on this slide. Please ensure that actual sampling time points, as well as the corresponding receptor sample concentrations that are corrected for dilution, are submitted. Similarly, examples of the data elements that may be included in the SAS PK datasets are included on this slide. Please note that the data columns such as treatment type, treatment or formulation group, donor ID, sequence, randomization code, replicate number, and unique cell ID are common between the PK and the concentration SAS datasets. Now, having covered specific considerations for the development, validation, and pivotal stages as relevant to the IVPT method, let us move on to some specific considerations for the analytical method. So over the next 2 slides, we will discuss specific submission considerations that are relevant to the analytical method validation studies. Please consider utilizing a suitable validated analytical method that includes a multi-point calibration curve, or CC, with suitable quality control samples, or QCs. Along with a detailed and well-organized information that is submitted in the analytical method validation report, consider providing summary details for each analyte based on data elements included in the bioequivalence or BE summary table 4 as they apply to your analytical method. So this BE summary table that I refer to are those that are utilized in ENDAs with in vivo PK studies, and they are available at the link provided below in the footnote. In addition to the elements from this BE summary table 4 as they apply to your analytical method, consider including additional data elements pertaining to working solution stability, drug stability in the receptor solution, brief descriptions of sample preparation and analytical methods, brief descriptions of the CC and QC preparation methods, as well as analytical method selectivity. Specifically for method selectivity, consider indicating whether interfering peaks were noted in blank receptor solution matrix exposed to the underside of skin in the presence of antimicrobial agents, if applicable. As a reminder, validation of the IVPT method and validation of the analytical method are separate and result in a separate set of reports and standard documents. Lastly, consider including adequate details of each relevant analytical method validation experiment in your analytical method validation report. Last but not the least, the next 3 slides include specific submission considerations as they pertain to sample analyses for IVPT studies such as method validation, including pilot and pivotal studies. Alongside detailed analytical reports, please consider providing separate tabular details of CC and QC summary information for method validation, pilot, and pivotal studies. You may include data elements from the BE summary table 14 as they are applicable to your analytical method. The link to the BE summary table is provided in the footnote. And consider including all IVPT runs that are relevant to IVPT method validation, including pilot, and to pivotal studies when you are calculating the precision and accuracy. It is important that the CC and QC concentrations are relevant to the range of the concentrations observed in the pilot and pivotal studies. Also, it is generally recommended that all samples from a given donor are analyzed in the same analytical run. Consider providing 100% numerical raw data including all original, re-injected, repeat analyzed, and reintegrated analytical runs as applicable, along with 20% serially selected representative chromatograms. For any samples and/or runs that were rejected, repeated, re-injected, and/or reintegrated, consider providing summary of the original value, the repeated, re-injected, or reintegrated value, as well as the reported value. Also consider providing detailed rationale explaining why the given sample and/or run was rejected, repeated, re-injected, or reintegrated, as the case may be. It is important that the identification and the reporting criteria for sample or run rejection, repetition, re-injection, or reintegration are objectively pre-specified in the SOPs that were active or effective at the time of the study. Now, analogous to the IVPT run summary details, as we covered in slide 25, for each analytical run associated with the method validation, pilot, and pivotal studies, consider providing summary details, such as, for example, analytical run number, analyte name, in case if there are multiple analytes evaluated, donor number, extraction and assay dates, instrument number, run status, and a brief description of the analytical run. So in summary, in this presentation, we looked at some important aspects of what information and data should be submitted in an IVPT submission, as well as how to organize the information and data that you submit. So our key takeaway from today is that it is extremely important that the IVPT submission is detailed, systematic, and well organized, such that the submitted information and data allows the assessor to adequately reconstruct how the submitted studies were conducted and verify the reported results. Please consider submitting high-quality IVPT submissions, as they can help potentially reduce the number of deficiencies, including those classified as major, as well as reduce the number of assessment cycles. For additional information, please refer to the appropriate agency guidances. Depending on the data that is submitted, additional information may be requested during the ANDA assessment. This presentation was a result of invaluable contributions from many individuals, as you see listed here. Folks from within the Office of Bioequivalence, as well as from multiple offices within the Office of Generic Drugs. So I would like to thank each and every one of them for their contributions. So thank you, everyone, for your interest and attention, and we look forward to your questions and feedback in the upcoming Q&A session.